Calcium accumulation in plants is a fundamental biological process that determines plant health, structure, and survival. Understanding how plants obtain and use this essential nutrient reveals the intricate mechanisms that support all plant life. Calcium, represented by the chemical symbol Ca is one of the most important macronutrients that plants require for survival. It serves multiple critical functions that are essential for plant health and development. Plants obtain calcium from the soil solution through their root systems. Calcium ions dissolved in soil water are absorbed by root hairs and transported into the plant's vascular system. Once absorbed by roots, calcium is transported upward through the xylem vessels along with water and other minerals. This upward movement is driven by transpiration. However, calcium has a critical limitation. It cannot move through the phloem, making it immobile once it reaches its destination. Calcium is fundamental for maintaining cell structure and integrity. As shown in this comparison, cells with adequate calcium have strong walls and stable membranes, while calcium-deficient cells become leaky and vulnerable to damage and pathogen attacks. Calcium exists in plants in several different forms, each serving specific functions. Calcium oxalate is the most abundant form, often stored as crystals. Calcium pectate strengthens cell walls, while water-soluble calcium remains available for transport and cellular processes. The most critical aspect of calcium accumulation is understanding that plants require a continuous supply. Because calcium cannot move through the phloem to redistribute from older to newer tissues, developing plant parts depend entirely on fresh calcium from the soil. This is why maintaining adequate soil calcium levels is essential for healthy plant growth and development. Calcium in plants serves far more complex roles than just providing structural support. It acts as a sophisticated intracellular messenger, coordinating critical cellular responses throughout the plant's life. Different environmental stresses trigger distinct calcium signatures within plant cells. Each type of stress, whether mechanical, osmotic, temperature, or oxidative, activates specific protein channels that allow calcium to enter the cell. Once calcium enters the cell, specialized calcium sensors decode these signals. These sensors, including calmodulin and calcium-dependent protein kinases, translate calcium concentration changes into specific cellular responses. Recent research reveals calcium's crucial role in plant defense mechanisms. Adequate calcium levels enhance resistance to pathogens like bacterial wilt in tomatoes and significantly reduce stem rot diseases in soybeans. Calcium also influences cell wall composition and structure. It affects pectin methylesterification, which determines cell wall flexibility and strength. During heat stress, calcium signaling triggers complex cellular responses that help plants adapt and survive. Calcium's multifaceted roles extend far beyond structural support. As an intracellular messenger, it coordinates responses to environmental challenges, enhances disease resistance, and regulates critical cellular processes that determine plant survival and adaptation. Calcium accumulation in plants depends on several critical factors. The most fundamental is soil conditions, which determine how much calcium is available for plant uptake. Soil pH plays a crucial role in calcium availability. In acidic soils with pH below 6, calcium becomes less available to plants. Conversely, alkaline soils may have too much calcium, creating imbalances with other nutrients. Soil moisture is equally important. Calcium moves to plant roots through water in the soil. During drought conditions, calcium uptake decreases significantly, even when soil calcium levels are adequate. Transpiration rate significantly affects calcium distribution in plants. Calcium moves through the xylem with water flow, so tissues with higher transpiration rates receive more calcium. This explains why young, rapidly growing tissues often show calcium deficiency first. These developing tissues have lower transpiration rates and depend on calcium redistribution, which is limited because calcium is immobile in the phloem. Calcium deficiency symptoms typically appear in young developing tissues first. These include leaf tip burn, stunted growth, and weakened cell walls that make plants more susceptible to disease. Interestingly, calcium toxicity can also occur, particularly in calcareous soils with very high pH. This restricts plant communities and can interfere with the uptake of other essential nutrients like iron and manganese. Foliar calcium applications offer a solution to calcium deficiency, especially when soil conditions limit uptake. Spraying calcium directly on leaves bypasses soil limitations and provides immediate availability to plant tissues. A fascinating example comes from tea plant research. Foliar calcium applications not only improve calcium nutrition, but also reduce fluorine accumulation in tea leaves. The calcium modifies cell wall structure and gene expression, creating a protective barrier against harmful fluorine uptake. This research demonstrates how calcium applications can serve multiple purposes, correcting deficiencies, strengthening plant structure, and protecting against environmental toxins. Understanding these factors is crucial for optimizing calcium nutrition in agricultural and horticultural systems. The future of calcium research in plants is rapidly evolving, with cutting-edge technologies opening new possibilities for enhancing crop nutrition and resilience. Calcium biofortification represents a promising approach to address nutritional deficiencies in crops. Scientists are developing multiple strategies to increase calcium bioavailability in food crops. Key biofortification strategies include genetically modifying calcium transporters, reducing compounds that block calcium absorption, enhancing natural uptake mechanisms, and improving calcium storage in the parts of plants we eat. Advanced genetic engineering and imaging techniques are revolutionizing our understanding of calcium transport in plants. This calcium signaling pathway diagram shows the complex networks that researchers are now able to study and modify. Modern techniques include CRISPR gene editing for precise modifications, fluorescent calcium indicators to track calcium movement, live cell imaging to observe real-time processes, and direct manipulation of calcium transporters. The future of calcium research lies in integrating massive data sets from multiple omic technologies. By combining genomics, proteomics, metabolomics, and transcriptomics data, scientists can gain a comprehensive understanding of calcium's complex roles. This integrated approach allows researchers to understand how genes, proteins, metabolites, and gene expression patterns work together to control calcium signaling and accumulation in plant cells. These research findings are being translated into practical agricultural applications. The images show various plant research studies examining calcium effects on plant health and cellular structures. Future agricultural practices will incorporate calcium-enriched fertilizers, improve soil pH management, precision agriculture techniques, and sustainable farming methods to optimize calcium nutrition in crops. Medicago truncatula serves as an important model organism for calcium research, particularly for studying how calcium oxalate crystals form in plant tissues, providing insights that can be applied to crop improvement. The future of calcium research in plants combines cutting-edge biotechnology with practical agricultural applications. Through biofortification, genetic engineering, and integrated data analysis, scientists are developing more nutritious and resilient crops that will help address global food security challenges.